Swayam Prabha. Digital India. Educated India.
Welcome back to Industrial Biotechnology course. Now, in the last lecture, I tried to discuss the glutamic acid fermentation process, is kind of amino acid production. So, we try to give you some kind of information that how the amino acid can be produced. Now, in this lecture, I am starting with new areas, that is production of penicillin, because the penicillin is considered as the first antibiotics that is ever produced, and that be used for curing the wounds, particularly we have during some accident and other things. So, that wounds is mainly due to the infection of gram-positive bacteria. So, with the invention of this penicillin, we can nowadays cure the disease. Particularly in case of World War II, it was very much required, because lot of soldiers was wounded and they required this kind of antibiotics for curing their wounds. And first question that is arises, what is called penicillin? Penicillin is a kind of antibiotics used to treat bacterial infection, because antibiotics, let me tell you, antibiotics it kills the germs actually, because it is kind of infection that we have, it kills that is antibiotics. And first naturally occurring antibiotics isolated from the mold. So, the mold is kind of fungi that is used for the production of these antibiotics, and discovered by accident 1928 by Alexander Fleming. So, this is a very famous scientist and who first discovered this penicillin, which has some antibacterial property. The penicillin antibiotics includes the ampicillin, the phenoxymethyl penicillin, amoxicillin, then first to second generation, fourth generation antibiotics that we produce from the penicillin. 50 drugs that are now classified as penicillin. The penicillin, I shall show you the speciality of the penicillin. It has the beta-lactam ring. So that we can produce 50 different drugs classified as penicillin. So use in World War II and after. So as I mentioned, this is after that it was used. Now this is, as I told you, this the penicillin, the speciality is that they have this beta-lactam ring that we have. It falls under the class of beta-lactam antibiotics. So this is called the beta-lactam antibiotics, and this is the thiazolidin, this is ring that we have. So most of the antibiotics, this portion is remaining constant. Only the acyl side chain, this will keep on changing, because this is the R group that keep on changing. That let me show you this. This is beta-lactam and this is the R group, and this R group might be different. We have if R group is this methyl benzene or phenyl benzene, methyl benzene, then we because this is precursor is the what you call phenyl acetic acid, we have phenoxyacetic acid, that we have different type of R group if present there. We have different types of antibiotics, but all are penicillin group, because all has the beta-lactam the ring that we have. So this is penicillin G, penicillin B, ampicillin, amoxicillin and methicillin. Now largely we use this penicillin G and penicillin B, because from penicillin G we produce ampicillin and other antibiotics. Penicillin V, the basic difference between the penicillin G and penicillin V is that penicillin G is quite unstable and acidic pH. So we know that penicillin usually taken in two different forms, either in the form of capsule or either in the form of injection fluid. Now, if it is injection fluid, our blood pH is almost close to the neutral. So, if you take a penicillin G in the fluid, we don't have any problem. But if you take as a capsule, our stomach pH is about 2. So, it is quite unstable at the pH 2. So, activity of the penicillin will be lost. So, we find comparatively penicillin G is better. So, penicillin G usually taken in the form of penicillin capsule. So, we have the classification of penicillin. We have natural or biosynthetic way. We can the penicillin that is harvested from the mold through the fermentation, we consider as a natural or biosynthetic penicillin V, penicillin G, and semisynthetic derived from the penicillin, because we make some kind of chemical alteration here to get the ampicillin, oxacillin, carbonicillin and ethicillin, etc., resistant to stomach acid. And this penicillin, they are resistant to stomach acid. We can take in the form of capsule, because then it would be quite stable. So, that is not natural, not pure natural, but it is semi-synthetic. Semi-synthetic means this is derived from the penicillin, which is mostly it is derived from the penicillin G. That is, it is so now question comes how the penicillin acts, on this organism which is pathogenic, because which causes the wounds. Because it's very interesting. It is resemblance protein needed to the cell wall. So its structure is quite similar with the protein needed for the cell wall formation. Penicillin binds with the cell wall of bacteria and prevent the peptide chain for linking and lyse it. So lyse it, you know that it is kill, it is just destroy that, and they do not interfere with the synthesis of other intracellular components, because they inhibit, for the main purpose of the penicillin, they inhibit the cell wall formation in the bacteria. So since the cell wall formation of the bacteria is inhibited, then cell wall will be killed. That is how the penicillin work, but it doesn't affect the intercellular component, because when we take in our system, it will affect the bacteria, but it will not affect the intracellular component of our system. So that is very important. These antibiotics do not affect the human cells, because human cells do not have the cell wall. Now this is how the penicillin works. This is, you can see how this is the cell wall synthesis inhibitor in growth media. This is the inhibitor that we have. They inhibit the synthesis of peptidoglycan, an essential part of the cell wall. The beta-lactam ring of the penicillin binds to the enzyme transpeptidase used in the formation of peptidoglycan cross-linking, and then the inability to form the cross-linking lead to the cell death. This is the reason why the cell death occur, due to osmotic imbalance. As soon as the cell wall has been removed, then what will happen? There will be some osmotic shock in the microorganism. Then the microorganism will be killed. This is because cell wall is a very important component of the microorganism. Now let us talk about a little bit about the history of penicillin. The first antibiotics were discovered in the year 1896 by the Ernest Duchesne, and he rediscovered by Alexander Fleming in 1928 from the filamentous fungus Penicillium notatum. So nowadays we produce penicillin by Penicillium griseus. I shall discuss how the transformation has been taking place. The first penicillin that has been identified for the formation of penicillin, that is Penicillium notatum, and this is how it grow on the surface of the petri plates, and the fungal looks like this, that whatever is shown in the petri on the petri plates. In 1928, Sir Alexander Fleming, a Scottish biologist, observed that Penicillium notatum, a common mold, has destroyed the Staphylococcus bacteria in culture. So Staphylococcus, we know it is a very harmful bacteria for us, and he found out that Staphylococcus, growth of Staphylococcus is greatly interfered, greatly inhibited in presence of this particular mold which produces the penicillin. It was first discovered by the Alexander Fleming like this, and then the sample of the molds from the contaminants plate he taken out, it was penicillin family, and later identified as Penicillium notatum. Later he identified the particular organism, what is the organism from the plate, and he identified the organism as the Penicillium notatum. The penicillin, the Fleming presented his finding 1928. He published the report on penicillin and its potential use, British Journal of Experimental Pathology. The production of penicillin during World War II importance is realized, as penicillin has been used to treat the many wounded the soldiers, because I told you before also this importance of the penicillin that visualizes after the World War II. Work of the production of penicillin, work by Alexander Fleming 1881 to 1955, Howard Florey 1898 to 1968, and Arts Chain 1906 to 1979 helped in the production of penicillin on a large scale for the first time for human consumption, that human use 1943. Now here I want to point out, in India, Sarabhai Chemicals, that is the one company that produce the penicillin through this fermentation process, and they use the Penicillium griseus chrysogenum for the production of penicillin. So now spectrum activity of penicillin. Penicillin is considered as a narrow spectrum antibiotics. You know that antibiotics has two type of antibiotics. One is narrow spectrum, another is wide spectrum antibiotics. Narrow spectrum antibiotics means they are very specific against any bacterial species, and wide spectrum antibiotics means they can act against the different type of bacteria. So that is as for example, that penicillin is considered as the narrow spectrum antibiotics, but streptomycin is considered as the broad spectrum antibiotics. And penicillin only can attack on the gram positive bacteria, and then some member like amoxicillin, because which derive from the penicillin, are also effective against the gram negative bacteria, but not Pseudomonas aeruginosa. The production of penicillin, let me discuss how the penicillin production take place. It is very it is common as compared to the particularly for citric acid fermentation process. You see that we have the lyophilized culture, then we know that how to activate the lyophilized culture. I told you that culture may be available in two different forms, either in the form of lyophilized culture or in the form of slant culture. The first, you have to activate the lyophilized culture and put it in the slant, because how it is done, it is very simple procedure that we have. So we know the lyophilization process. I already discussed. Lyophilization is a freeze drying process under frozen condition. This is the moisture is taken out, the water molecule from the solid to vapor phase is taken out, so that your microorganism can be preserved for longer period of time. This dry powder, which contain the cells, which can be preserved for couple of years, we can take it out, and then we make a suspension in 0.85% saline water. Just we can make a suspension. Then we can take one spoon and this one loopful of this culture, and then we stick on the particular slant, that agar slant. We can have that. We have media. We can put the sketch on the surface. So while we produce this slant, this is slant, and then agar slant, and then we this agar slant, then we from this we keep it in the incubator, allow the cell mass to grow. After that we take this culture, we transfer to the liquid media, which is in the conical flux. Conical flux we have liquid media. So this is from this culture we transfer here. When grow we transfer here. So here what is then we take it in the inoculum vessel. We increase the volume, and finally we put it in the inoculum vessel. I told you the volume of the inoculum vessel is 5 to 10% as compared to fermenter. Now here I want to point out that in case similar to the citric acid fermentation process, that we use Aspergillus niger, which is also fungi, and here we use Penicillium chrysogenum, which is also fungi. So what is happening that we shall have this fungi is the filamentous organism. So I shall show you the structure, morphology of this particular organism. That what we will observe that this organism we cannot count. So what we have to do, we shall have to put do the sporulation of this microorganism. And sporulation, I have already shown you how the sporulation is done. I mentioned that by using the weighted these barley grains and honey peptone media, it is possible to produce the spores of any kind of fungal cells. So, and here I want to mention that Penicillium chrysogenum, this has a green spore. Green spores, that is so in case of this what you call in case of Aspergillus niger, we had the black spores, and here we have the green spore, and we can count it and we can prepare the spores for culture in the lab. And this culture we take it in the seed can, and from the seed can we transfer to the inoculum vessel. This is what we call pre-fermenter. And now here we produce the vegetative cells, and these vegetative cells you put it in the production fermenter. And after production fermenter, first you pass through the filter. Filter means rotary vacuum filter, because the rotary vacuum filter already I explained, since the fungal size are quite big, couple of millimeter, so it can be easily separated with the help of rotary vacuum filter, where you can get the mycelium, and this mycelia can be used for the production of rough quality paper. Then you put it in the cold tank, and then to the purification and recovery process. This is the steps involved in the penicillin production. A media is corn stem liquor, is the byproduct of the starch manufacturing industry. The corn stem liquor that we have, and it is considered as a good nitrogen source. Then we have yeast extract. This is also considered the good nitrogen source, and other substance added to the. I mentioned you before that two type of media that we have. We have complex media, we have synthetic media. Complex media means when the composition of the media is not well defined, but whenever you use any kind of natural product, corn stem liquor is a kind of natural product, is produced from corn, and so this and yeast extract also comes out from the yeast, and other substance added, particularly these two substance, if we present any of these two substance present in the media, media, we can consider the media as a complex media because the composition cannot be defined properly. And after 40 hours, penicillin begins to be secreted in the fungus. So the mold mycelia is filtered from the harvested product, and the penicillin is extracted from the organic solvent, butyl acetate or amyl acetate, in which it dissolves, and potassium salt are added in penicillin precipitate to form that is was and dry. Let me tell you the things how really it happens. So in the penicillin production, because after the fermentation is over, fermentation takes about 5 to 6 days. After the fermentation is over, we take out the fermentation broth and we separate out the cell by using rotary vacuum filter, then filtrate. We take it out, filtrate contains the penicillin. So penicillin is recovered by using the solvent extraction process, and solvent we use the butyl acetate. The butyl acetate, now this is very critical process, because in the lab you have Buchner funnel. With the help of Buchner funnels, you have two different type of liquid solvent we use. We can shake properly, so that with the respect as the solubility at the partition coefficient of particular solute is different in the different layer of solvent, so it moves from one layer to other layer. But industry, we are handling huge amount of liquid. The question comes how the penicillin present in aqueous layer goes to the solvent layer. So they mix they have some kind of mixing like this. They have through mixing they mix in such a way both the layer they can mix thoroughly, and so that one layer is one solute present in one layer goes to the other layer. But solubility depends on the pH of the media. We found that at acidic pH, the solubility of penicillin is more in the solvent layer, and in the neutral pH, solubility of penicillin more in the aqueous layer. So when we use the acidic pH, then your penicillin will go to the solvent layer. Then these two butyl acetate is insoluble in water, so you can easily separate the aqueous layer from the solvent layer. And aqueous layer, what is considered as a industrial effluent, you have to discard it. Again, you use some water and use some kind of buffer solution, or so that maintain the pH as 7, so that when you again mix together, then your penicillin will come back from the solvent layer to the aqueous layer. So then again you take this solvent out, again purify the solvent, again you decrease the pH to 2 by the help of acid. Then again you recover the solvent in the penicillin in the solvent layer. Like this you can purify, because penicillin is to be purified properly, because penicillin, as I mentioned, it is usually marketed in two different forms, either in the form of capsule or in the form of injection fluid. If it is the injection fluid is the 100% free from the contaminants. So your fluid should not have any kind of contaminants present, because as soon as you inject your bloodstream, if any contaminants is there, your health immediately will be affected. So there is no contamination. Should the 100% sterility you have to maintain. But in case of capsule, that 100% sterility may not be required, because as I told you that when you take in the form of capsule, it goes to the stomach where your pH is 2. If some microbial infection is there, then your system will take into account. Then finally you use some kind of potassium salt. Potassium salt might be potassium acetate or potassium carbonate. We add so that potassium or sodium salt of penicillin can be separated out. And finally we do the crystallization process. Through the crystallization process, the penicillin salt or sodium salt, we take it out from the system. Now, the penicillin was the first important commercial product produced by aerobic submerged fermentation process. First antibiotics to be manufactured in bulk and used as input of for some semi-synthetic antibiotics I mentioned before. It is fermented in a batch culture. A fed batch process is normally used to prolong the stationary phase, so increase the production. Here let me tell you that particularly in the penicillin fermentation process, through the fermentation process we produce the beta lactam group that is produced, but actual production of penicillin depends on the precursor. We have seen the R group that is different for that the different type of penicillin. So we have in the fermentation process we usually produce two type of penicillin. One is penicillin G, another is penicillin B. For the production of penicillin G, we use the phenyl acetic acid. Phenyl acetic acid, and this is in case of penicillin G. And in case of penicillin B, phenoxyacetic acid. Penicillin B. So we use this two different precursors. This is the precursor that is required for the penicillin fermentation process. So so this is the profile of the different component present in the reaction mixture. The lactose that is used as a carbon source, this is keep on decreasing with respect to time, and you can see that biomass production is increases like this, and penicillin production increases like this, and ammonia that considered as a source of nitrogen, it decreases like this. Like all antibiotics, penicillin is the secondary metabolites. What is the secondary metabolites? I told you that when organism, we have seen that life cycle of the organism, that this is the viable cells, this is time. So when you plot that, it is like this. So we have lag phase, you have log phase, you have stationary phase, you have death phase. This is called stationary phase. Now this stationary phase, that what you have, that we have this that this is called also starvation phase. This is called starvation phase. So starvation means we don't have sufficient substrate for the organism, so organisms struggle for survive. That is the difference we have. And like all antibiotic, and during this process their metabolism little bit changes in that. So penicillin is produced there, that is why we called it as a secondary metabolite. So it is only produced in the stationary phase. It is only produced when the growth of the fungus is inhibited by stress, and it is not produced during the active growth phase. What you call here, if you look at, this is considered as the active growth phase, but here you see that when the organism almost going to the stationary phase, then your penicillin production started like this. So that is why we call it, the penicillin is considered as a non-growth associated product, because the rate of production is proportional to the cell mass, but this occurs only when the organism at in the stationary phase. Thank you very much.